Okay, we're going to switch up a bit here and step away from talking about animal cells or eukaryotic cells in general, and we're going to see what there is about plant cells in particular. Excuse me. Plant cells. All plant cells are going to have some form of a cell wall. Okay, cell wall does three things. Primarily support. Okay, you get a tree, a sequoia tree that's 200-300 feet tall. That's a lot of weight. That's tons and tons and tons of weight pushing down. Well, you got to have some structure there to be able to withstand all that downward pressure due to gravity and weight. Cell walls. Got to keep stuff in, keep stuff out. That's what this barrier to infection is all about. Keep out bacteria, keep out viruses, keep out some pests, insects, rodents. And we're going to see later in this lecture, also going to give little windows to be able to cells talk back and forth to each other. We call these windows plasmodesmata. Now, this is different than us animals. Us animals, we use collagen and other fibrous proteins. Cells are going to be stuck together, and at the end of this lecture we'll talk how animal cells stick together. Well, you get a clump of cells, and then you get another clump of cells, and you want those to stick together, so what you do is you wrap stuff around them. Collagen and some of these other protein strands will wrap around them, and then get a bunch of those, bundle those together, and you've gone from a single cell clumped with other cells to a tissue. Collagen, some of these others will then wrap around tissues, and you get an organ. Now, when it comes to cell, how they stick together, primarily that's going to be these two right here. Glycoproteins and proteoglycans. Oh, I love these words. Remember, rule of thumb, if you don't know what it is, cut the word down into its components. Let's see. Glycoprotein. Glyco means sugar. So these are proteins that have sugars added to them. You know, not just one sugar. Here's a glucose, here's a glucose. No, it'll be long chains of sugars. So at the base, at the center of this will be a protein, a polypeptide, or a group of polypeptides forming the quaternary structure, and then added onto these at specific amino acids will be sugars. 1, 2, couple of dozen, whatever. On the flip side are what is known as proteoglycans. Protein, glycan, glyco, sugars. So this has long strands of sugars, a carbohydrate, that has some amino acids attached to it. Okay, keep in mind, glycoproteins, a protein with sugars added to it, proteoglycans, a carbohydrate with some amino acids. This is going to hold cells together. This is going to allow cells to talk to each other. Now, when it comes to intercellular junctions, plants, as you see here on the left, have it easy. Why do they have it easy? Well, because of this cell wall. The cells have been removed. All that's left is a cell wall. Cell wall. It's like brick and mortar. You lay down a brick, cover it with mortar, lay another brick on top of it. Mortar dries, the two bricks are now glued together. Same with plant cells. It's going to be different though When you look here with animal cells, no cell walls. No cell walls anywhere in your body. So, you have to have a different way. Well, we're going to have those proteoglycans and glycoproteins, and all those things are going to glue the two cells together. They're going to span across. They're going to hold the plasma membranes together. Now, because plant cells have that cell wall and they're glued together, that cell wall can be somewhat impenetrable, hard to get things across, hard for neighboring cells to talk to each other. So, evolution's found a way around this. Windows. A window from cell A to cell B. We call this window a plasmodesmata. Plasmodesma, singular. All the plasmodesmata allow for the interplay, the exchange of cytoplasm and nutrients back and forth between neighboring cells. So instead of having to pump whatever it is out via a vesicle, have it float next door, and then somehow be pulled in, now they can just swap back and forth. Which is going to be a lot different in animal cells. All of our cells are squishy, no cell walls. So we have to have some way to, since we can't glue cell wall to cell wall and have this impenetrable defense, we've had to evolve other mechanisms. One of the mechanisms we've evolved to utilize is what is known as tight junctions. Tight junction. You have over here cell A, this is the plasma membrane for cell A, over here cell B, and its plasma membrane to the right. One of the junctions we have is what is called tight junctions. Tight junctions are literally, as you see right here, where the two plasma membranes are pinched together. They're not fused, but they're pinched together and held there by proteins. Proteins that start in cell B, cross out, and then back in into cell A. Back and forth, kind of sewing them together. What's so important about this? Well, this tight junction may be between cells lining your stomach or your large intestine. Fluid comes, hits, and then bounces right back out. So that way you are not leaking stomach contents. You're not leaking intestinal contents. It's like a Ziploc. Okay, it holds the stuff in, keeps stuff out. You do not want those digestive enzymes sloshing around in your gut, because then you'd start digesting your own organs. So, tight junctions, watertight fit. Nothing leaks. But you need to hold cells together. You need to hold cells together so you have, well, pretty tight tissue. Think about it. Go pull on your skin anywhere. It takes a lot, it doesn't take much to stretch the skin. You can pull on it. But it takes a lot of physical force to be able to tear the skin, to separate the cells within the tissues. Same with your muscles. Same with, the cells within the tissues of your gastrointestinal tract. We can't glue them together, but what we can have are proteins like the desmosomes that start in cell A and then pass through, kind of like rebar holding two sections of concrete together. This joint, well, it's not a tight joint, it's not leak proof, but it is tear proof, it is separation proof. Our cells also have their own little windows. They're not desmata, Those are permanently open windows between plant cells. What we have are called gap junctions. Little protein structures that open and close, kind of like the nuclear pores from an earlier lecture. Same thing here. They open, things rush in, things rush out of the cells, between the cells, and then they close. Why? Well, again, under normal conditions for two cells to exchange, cell A has to pump stuff out via vesicles through the plasma membrane, and then it has to happenstance float by cell B, and somehow cell B is able to capture it in a receptor or phagocytizes it, pulls it in, and that takes time. Whereas these gap junctions, relatively quick. They open, they close. When they're open, stuff rushes from A to B, B to A, back and forth very, very quickly. Allows for not instantaneous, but very quick exchange between neighboring cells. So instead of something being measured in minutes and hours, if it has to be made, pumped out, and hopefully brought up by the neighboring cell, now we've got it minutes, seconds exchange through an open or close, open gap junction.